So let us see what are the different kind of deficiency symptoms that are shown in the plants. There are four major type of deficiency symptoms. The first symptom is chlorosis. The second symptom is necrosis. The third symptom is stunted plant growth, and the fourth symptom is premature fall of leaves or premature fall of buds. So these are the four major type of deficiency symptoms that are most generally seen. Now, if we see another fifth variety which is not visibly appeared, matlab you can't see it visually, but it happens is the inhibition or the stopping of cell division. So let us see each symptom in detail. When we talk about chlorosis, it literally means yellowing of leaf. You can see in this diagram where one leaf is completely becoming yellow in comparison with the other leaf which is green in color. Now this is due to the loss of chlorophyll. Chlorophyll is the pigment that gives plants the green coloration. So without chlorophyll, they lose their green color and become yellow. Now, which deficiency deficiency of which elements can show chlorosis? Chlorosis can be shown when there is deficiency of nitrogen, potassium, sulfur, iron, manganese, zinc and molybdenum. Okay, so all these elements deficiency have one common symptom, that is chlorosis. The next one is necrosis, which is nothing but death of a tissue. If you can see in the diagrams, there are particular areas in this leaf which are completely dead. If you can see, one part of the leaf is dead. It became brown in color. It is dead part of the leaf. So like this, there is death of the tissue. And this happens due to the deficiency of calcium, magnesium, copper and potassium, and this is called as necrosis. The third one is lack or low level of nitrogen, potassium, sulfur and molybdenum. It causes cell division inhibition. Now cell division inhibition, even though visually you can't see it, you can see an overall symptom of it by stunted growth. Now, if cell divides continuously, especially in the apical buds, then only the plant will grow tall. If the buds are not dividing properly, if the meristematic areas in the cells are not dividing properly, and if there is an inhibition of cell division, then what happens is there will be no growth, and stunted growth is an indirect result of the inhibition of cell division. Now, some elements like sodium, potassium, molybdenum, what they do is, if their concentration is low, they inhibit flowering or delay flowering. Let us say flowering has to happen in this season, let us say in July, and it should start at the first week of July. The flowering may start somewhere at the third week of July. Or even if it starts, the bloom does not stay for a very long time. All this happens due to the deficiency of nitrogen, sulfur and molybdenum. There is another set of micronutrients also whose deficiency can cause various diseases. Zinc causes mottled leaf. So this is a mottled leaf image where you can see clearly there are bronze colored spots on the leaf completely. This is due to zinc deficiency. And it is characterized by chlorosis also and reduced size of the leaf. The leaf becomes very small in size. Copper causes a disease called dieback of citrus. You can clearly see that condition where woody plants, the peripheral parts of the woody plants, that is the side parts of the lateral branches, the leaves, they all get killed. So this is called as dieback in citrus, which is seen in the citrus varieties, which is due to copper deficiency. The third variety is called boron deficiency, causes heart-rotten beets. As you can see there, in the middle of the beetroot, there is rotting that is happening here. That is called as the heart-rotten beets, which is due to boron deficiency. Molybdenum deficiency causes whip tail in cauliflower. Cauliflower, actually you have a big flower with a bloom and a very huge broad leaf. But here in this image you can see the flower itself is very small. The leaves, instead of being broader, they are now longer in length. So this is called as the whip tail in cauliflower, which is caused by molybdenum deficiency. Nickel causes the mouse ear leaf in pecan. Now pecan is a nut plant. If you can see on the right side you have the normal leaf of pecan, and on the left side you can see the mouse ear. Very clearly you can see the shape of the leaf got altered. Normally it is like this, whereas in the altered shape it has become into an into a kind of an oval shaped structure which is very similar to the ear of the mouse. That's why this is called as the mouse ear leaf disease in pecan. Okay, and chlorine results in this bronzing in legumes. The leaves of the leguminous plants show bronze-like appearance. So these are the various deficiencies of mineral elements and the symptoms showed by various plants with respect to deficiency. Now let us talk about toxicity. We also understood that toxicity is also due to excess amounts. Now, if there is a moderate decrease, it causes deficiency. If there is moderate increase, then it causes toxicity. We've already understood this in the previous video where we were talking about this critical concentration graph, where there is the minimum concentration and the maximum concentration. If the nutrient amount have a decrease from the minimum concentration, it causes deficiency symptoms. If the nutrient concentration increases beyond the maximum critical concentration, it causes toxicity. So in other words, we can say that beyond the optimum level, so this is the graph once again that we spoke about. So this is the critical concentration. This is the optimal level. So this is the minimum threshold. This is the maximum threshold. So whenever there is the cross of this maximum threshold, you get toxicity. And whenever you cross the minimum threshold, you get deficiency. So this is the optimum level, and this is a very narrow range. You cannot play much with the range of the optimum level of concentration. Okay. Now, any mineral ion concentration in tissues that reduces the dry weight of the tissues by 10% is considered toxic. Such critical concentrations vary in nutrition. So what is dry weight? When you remove the entire water, let us say I have a leaf and the leaf weighs 1 gram. I sun dried for 2-3 days. Again I weighed. The leaf is only 300 grams. So the 700 grams was that of the water. So if dry weight means when you remove the entire water content from a tissue and weigh it, then it's called dry weight. So if there is any mineral element that is decreasing the concentration of the dry weight of a tissue by 10%, then that element concentration is toxic concentration. Okay, so toxicity symptoms are difficult to identify. In fact, toxicity levels also, they vary from one organism to one plant to another. In fact, it was found at a lot of times that one element, if it is in toxic concentrations, that is if it is excess and it is becoming toxic, it inhibits the uptake of the other element, another element. So what happens? You might not show the symptoms of toxicity, but you might show the deficiency symptom of the other element, whose uptake is inhibited by our toxic element. For example, take the case of manganese. Now, manganese toxicity you can very clearly see by these brown spots that are seen on the tips of the leaves and the chlorotic veins. If you see the veins here, let me show it to you. If you see the veins, they are all completely yellow. The area around surrounding the veins, they have lost the chlorophyll, and you can see these brown spots. So these are the symptoms of manganese toxicity. But if you understand it more clearly, you can say that manganese is in a competition with iron and magnesium. So what it does, it always competes with iron and magnesium. Okay, so what happens is when manganese, it inhibits the translocation of calcium to apex. First it causes deficiency of magnesium, and it binds with and it binds to the enzyme where magnesium is supposed to bind. So without magnesium, what happens? There will be no chlorophyll, as a result the chlorosis. Secondly, manganese also inhibits calcium translocation to shoot apex. Means what it does, it will not allow the calcium to move to the tip of the shoot. If calcium does not move to the tips of the shoot apical meristem will not get enough calcium to carry out cell division. So as a result, there will be inhibition of cell division and stunted growth because of calcium deficiency. So manganese, by becoming excess, is causing deficiency of magnesium and calcium. So the symptoms we see can be of the symptoms which of deficiency of calcium and magnesium instead of excess manganese. So therefore, excess manganese induces deficiency of iron, magnesium and calcium, which appears to be the symptoms of toxicity. What we consider as magnesium toxicity symptoms are actually symptoms of the deficiency of iron, magnesium and calcium created due to the excess of manganese.